So John mentioned the PI3 kinase inhibitors, and that perhaps one of the appeals of them is that they have a different mechanism of action. They're not the cytotoxic chemotherapy that we've been using. Matt, so can you explain that a little bit?
Yeah, so it's been an interesting story to watch the landscape of the PI3 kinase class kind of take shape, starting with the the delta inhibitors, and then you have the delta gamma inhibitors, and now you have delta alpha inhibitors, and so these isoforms, at least the delta specific, isoforms felt to be the pathway that was involved in some ways, the B cell pathway and lymphomagenesis in regards to that. Now the contribution that the alpha isoform brings for potentially drug resistance, but also looking in bringing in the gamma and where that may play in certain types of lymphomas. And so I think, when you have now, 3 agents, what you're looking at with approvals in the follicular lymphoma space, kind of how you're gonna use them moving forward is gonna be interesting, and to kind of slice and dice, if you will, those drugs.
Right. So there's these 4 different isoforms, and I remember originally when we were working on idelalisib, the concern was that by using one isoform specific inhibitor that you would get mechanisms of escape through some of the other isoforms. And I think there's some data at least in mantle cell lymphoma, that's a real possibility. Less clear to me in some of the others. At least it's hard to prove one way or the other. But I guess 2 FDA approved PI3 kinase inhibitors for relapsed follicular lymphoma, both idelalisib and copanlisib. They are different. What do you think of these? Do you think it makes a difference? One's a pure delta inhibitor. One's also inhibits the alpha isoform. What are your thoughts?
Yeah, so idelalisib, as you said, is the first one that was approved, and that hits delta specifically, and it's an oral agent that is approved for relapsed and refractory disease. And I think the study that really showed its efficacy was a really strong study in terms of looking at probably the highest risk people with follicular lymphoma, this double refractory group of people that have refractory disease to both alkylating agents and rituximab. And there you see a really nice activity and efficacy profile, but the challenge, as we've learned over time, is that there is also a unique toxicity profile of transaminase elevation and some colitis that can be very severe, and pneumonitis. And there's also infection. So, as an oral agent, it's appealing, but there are these toxicities that have to be considered. Copanlisib came along, I think, to 2 important differences from idelalisib. One is that it also targets the alpha isoform, so you get alpha and delta inhibition. The second major difference is that it's given intravenously, and the schedule is an hour once a week for 3 weeks out of 4. So it's a little more of an injection burden for patients. But in terms of the Chronos study that you just referred to, it shows that the response rate is somewhere close to 60%. CR rate is low. It's probably on the 13 to 15% rate, but it has a very different toxicity profile. So you don't see the same transaminase elevation, but what you do see are more of the alpha effects. So you see hypertension and hyperglycemia that's very transient. And I think for people who are not expecting it, it can be probably pretty shocking, because the hyperglycemia occurs very quickly after the infusion and can be quite high, but then also rapidly drops. And same thing with the hypertension. You can have, grade 2 or more hypertension, but again, it drops, and most people tend to be asymptomatic. So I think using copanlisib just needs some awareness of what toxicity to look for. All of that being said, I think the toxicity profile is favorable. These are transient effects, and I think whether or not you use one or the other probably is going to have more to do with reimbursement and patient selection in terms of an oral versus IV than anything.
They do have very different profiles, and I guess one adverse event profile. So I guess one explanation is that the it's the different isoforms that we're hitting, right? So we're hitting delta versus delta and alpha. I guess other people have argued, and some of the authors on the copanlisib study had argued that it was the schedule, and by not constantly hitting it. John, you've done a lot of work with PI3 kinase inhibitors. What's your take on that? Do you think it's, I guess I'm not so sure that any of the drugs that we're giving, we know the right dose and schedule, and maybe we should be exploring some of this other schedules with some of the drugs that we already have.
Yeah, I think it's a fair point. I think that at the end of the day, we don't have a great sense of whether or not changing the schedule, changing the exposure to the drug will, it'll probably ameliorate toxicity, certainly, but from the standpoint of compromising efficacy, we don't know. I think that the toxicities for both of the PI3 kinase inhibitors that are improved, I think, the good part is we know, and I think we were a little surprised, or some people were surprised with idelalisib, with what occurred as far as toxicity. I think now we know, what can happen, and I think if we're monitoring patients, the expectations are different. And I think with copanlisib, there are some, pretty well-established monitoring guidelines and some predictability as to kind of when you have to worry about the toxicities and then when you get beyond them. So I think, in my mind, these are particularly useful drugs for older patients, and we have a lot of follicular lymphoma patients that are older that, when they've been through a couple different chemotherapy regimens, they're not excited to have them, they're not headed to a transplant. You can get good mileage, and again, with care and with monitoring, can manage the toxicities reasonably well.
I guess we should also talk a little bit about idelalisib. So idelalisib is a PI3 kinase delta and gamma isoform specific inhibitor, so it doesn't really pick up much in the way of alpha. That's currently in development. This is a drug that has been studied in a variety of hematologic malignancies, including low-grade lymphoma. In the Dynamo study, we saw about a 50% overall response rate in that very difficult patient population that you talked about earlier, where that was the patients that were double refractory to both rituximab and to chemotherapy. And so I thought that was pretty exciting data. We know that will hopefully be, that's before the FDA, should be by October that we should know whether that one gets approved. It does have some similar toxicities in that it has, that we see perhaps with idelalisib. It has an incidence of transaminitis, the problems with late problems with colitis, and of course with all these different drugs we worry about infectious complications. But I look forward to seeing whether that gets approved soon. I think that it's the other thing that might be exciting outside the role of follicular lymphoma, but by inhibiting gamma, perhaps we're going to extend the number of diseases that can be treated with it, perhaps into T-cell lymphomas and other malignancies.